This conference will now be recorded.
All right. Good morning, everybody. Welcome to another EcoFoci seminar series. I'm Emily Lamacci. I'm co-lead of the seminar series with Deanna Krauser. And this seminar is part of NOAA's EcoFoci biannual seminar series focused on ecosystems of the North Pacific Ocean, Bering Sea, and U.S. to improve understanding of system dynamics and application of that understanding to the management of living marine resources. Since 1986, the seminar has provided an opportunity for research scientists and practitioners to meet, present, and provoke conversation on subjects pertaining to physical and fisheries oceanography or regional issues in Alaska's marine ecosystems. Visit the EcoFoci webpage for more information. And we sincerely thank you for joining us today as we transition to this hybrid, virtual, and in-person fall seminar series. We look forward to the speaker lineup you can find at the One NOAA seminar series and on the NOAA PMEL calendar of events. And if you missed the seminar, you can catch up on PMEL's YouTube channel. It takes a few weeks to get these posted, but all of the seminars that are recorded will be available there. And I ask you to please make sure that your microphones are muted and you're not using video. And during the talks, please feel free, if you're online, to type your questions into the chat. We'll be monitoring for questions, but we will wait to address them until the end. And today I'm excited to introduce three speakers, Deanna Krauser, Allison Drury, and Ned Coquelet. This is a trial of a new 5-minute lightning talk format for the EcoFoci seminar. We also have full seminars and paired 15 to 20-minute talks that we'll be trying this fall series. Today, we'll have the opportunity to hear from multiple speakers about recent projects, ecosystem indicators and measurement techniques, and more. And this session will begin with an introduction to the ABT communication method and end with an open discussion session with our speakers as a panel. I'm going to begin with Deanna Krauser. Deanna is a zooplankton ecologist and a contractor with Linker Technologies in support of NOAA's Alaska Fisheries Science Center. Her research focuses on how environmental conditions affect zooplankton size and distribution.
Thank you, Emily. All right. So as Emily said, my name's Deanna Krauser. I'm a zooplankton ecologist here and here at NOAA, and I have spent the last 2 years in a communications training program specialized for climate change. Climate ambassadors are passionate and dedicated aquatic scientists that want to reach a variety of audience and expand their thinking on the impacts that humans have on our aquatic environments. And we do this through use of communication.
This conference will now be recorded.
Oops. And we do that through a use of a variety of communication tools learned and practiced over 2 years' time. To learn more about the program, just search AFS Climate Ambassador Program. And before our lightning talks, I just want to spend about 10 minutes teaching you a little bit about what I learned in hopes that I can contribute to your science communication skills and add new tools to your toolbox. So one of my favorite tools that I learned in the Climate Ambassador Program was this ABT method, which is a storytelling structure developed by a marine biologist turned filmmaker, Randy Olson. Though its origins reach back thousands of years to Aristotle and the beginning of narrative culture, the ABT was used in the Gettysburg Address and in MLK's I Have a Dream speech. Even the scientific method is built around the ABT, which underlies the IMRAD narrative template for scientific papers. I found the ABT to be one of the most compelling arguments for using a good narrative form while communicating your science. So I'm going to start this talk by talking about storytelling. So there was a researcher, his name was Yuri Hassan at Princeton's Neuroscience Institute, And he looked at brain activity while subjects watched 4 different types of movies. The first movie is in green to the far left, and it was a suspense murder mystery. Bang, you're dead. And it was jam-packed with plot twists and foreshadowing. The second movie subjects watched was in blue, The Good, the Bad, and the Ugly. It was a less suspenseful Western movie. The third, in red, Curb Your Enthusiasm, was a sitcom. And the fourth, in orange, down at the bottom right corner, was a Washington Park reality segment. So it was just people randomly going about their day. To quantify the differences, we can look at the ISC score for each movie. And the ISC score is how they quantify brain activity. So you can see in the green, it's the ISC score is much higher for the murder suspense mystery movie than compared to orange to the far right, where subjects were just watching random people walk around in a park. So what is the difference between the 4 movies? Well, the answer is narrative. Narrative is a series of events that occurs in search for the solution to a problem. And the but is the powerful word of contradiction that changes the direction. It fires up the brain. You identify the problem when you say but. So if you look at the difference between these 2 scans, the one on the left is the reality segment. It had a non-narrative. And the one on the right is the high suspense murder mystery, and it had narrative. And this is what you want. You want your audience to be engaged. You can tell that from the brain scans that people were sitting on the edge of their seats. They were actively listening, actively trying to follow the story, actively trying to predict the plot. That's what you want when you're communicating your science. You want everyone to be as engaged as you are. And that's what the power of narrative brings you. And this is just a setup of why it's so important to use narrative when we're communicating our science. So there are 2 types of bad narrative forms, which we have to try to avoid if we want our messages to stick when communicating our science. The and, or AAA, is one of them, and it has too little content, too little narrative. There's no contradiction, which means that there's no narrative content. Now, our inner circle can listen to this because they're just as interested in your work as you are, and it's just an information dump, really. So you can do this to your inner circle, but if you were to and an outer circle, no one's going to really be able to follow what you're saying. They're not going to really be involved. They're not going to understand. And so this is another a type of bad narrative form. The other one is despite, however, yet, which has too much narrative content. It has multiple contradictions. And this is something that we also want to avoid. This just causes confusion. A good narrative sits right in the middle. And, but, therefore. It's the optimal amount of narrative content to keep your the people who you're talking to engaged. This is how you reach your outer circle, and this is when narrative is important, when you're telling a story. As scientists, we're used to communicating our science, and we are familiar with using a narrative template like the IMRAD. But not everyone here is familiar with using a simpler storytelling structure for everyday use, like outreach, presentations, proposals, and more. Therefore, I would like to introduce you to the ABT method. So this is breaking down the ABT. And is your setup, it's your agreement. It's literally the introduction for your scientific papers. Your but is your tension, your problem, the contradiction, confrontation. It's your methods and your results. And then your therefore is your resolution, your solution, your consequence, your action, your discussion. So sorry, I'm sick a little bit, so bear with me. The IMRAD follows a basic ABT structure, but there are some major differences in how people outside academics absorb information. Therefore, we need to adjust our communication approach for the outer circle. So here's an ABT, and you can see that the IMRAD follows a basic ABT structure. That's our setup. There are some major differences in how people outside academics absorb information. There's our problem. Therefore, we need to adjust our communication approach for the outer circle. So this is our solution. So it's a simple 3-sentence setup in which you can, it could really, an ABT can be as long as you want it to be. It could be 3 sentences. It could be an entire presentation. It could be a whole movie. You know, that's the best thing about an ABT is, you know, it could be as big or as little as you want to, but this is its simplest form. 3 sentences, but therefore set up a problem and a solution. Now, one of my favorite tools that I learned for the ABT is using a past and present and future setup. So basically, ABT goes like this. In the past, life was like this and all was well. But now we have this problem. Therefore, in the future, we're going to do this. So this is how we set up our ABTs. But I'm sure you guys, I'm just giving you some examples and breaking it down for you. But sometimes it feels hard to start. So we'll have to see, the first step is identifying your problem. When you identify your problem, you have to consider the following. Do you have a single narrative? When we're creating an ABT, we don't want to have multiple narratives that are contradicting each other or confusing anyone. We want it to be simple. We want one narrative. You also have to make sure that you have the correct source. Is the problem pollution killing fish in a river, or is it laws that allow for pollution? What's preventing the solution? If the problem is pollution, then why isn't it stopped? Step 2, the and. The and material has 2 main elements, the what and the why. Your what is a simple opening description in an ordinary world, like this. Management of caribou herds in Alaska has been studied for over a century. Now, your why is the why should we care element. What's at stake? And caribou are a major source of Alaska's 126 million annual income from tourism. Now, you should end the opening of your and statement as peaceful. It should be an end of story feeling. So if you put it together, management of caribou herds in Alaska has been studied for over a century, and caribou are a major source of Alaska's 126 million annual income from tourism. I feel like that's the end of the story. I feel like it's peaceful. I feel great. Here's a note always to keep in mind. The power of storytelling always rests in the specifics. So try to make what's at stake element as specific as possible. So not just caribou are important, but actually how they are important, tourism. And if possible, add a dollar or an aesthetic value. The but and therefore. Now these are 2 moments. The 2 moments of transition are the best chances to break through the noise and capture someone's attention. You want to maximize contrast by going from calm, when you felt like at your and, you feel like the story is calm, peaceful, it's resolved, you heard the end of the story, everything's fine, to excited. You want to go from the what to the how and not in reverse. So the but begins the contradiction with what the problem is and then follows with the how of the problem. So the right way to do this would be caribou are threatened with 90% population loss in the next year from habitat loss, pollution, and hunting. The wrong way would be, but habitat loss, pollution, and hunting now threaten caribou with 90% of pollution in the next year. So you set it up with the overall statement, and then you talk about the process that causes it. I mentioned earlier that the ABT was used in MLK's I Have a Dream. So I wanted to share that just to prove to you that this has been used, and this is one of the most famous speeches that we have. Well, I mean, there's a lot of famous speeches, but this is a very, very famous speech. And so we can go over and break it down. Your and is in the blue, your but is the red, your therefore is the green. I'm going to paraphrase it a little bit. You got your 5 score years ago. It opens with time, which is one of the most powerful aspects of a narrative. A great America in whose symbolic shadow we stand today signed the Emancipation Proclamation. And this momentous decree came as a great beacon light of hope to millions of Negro slaves. who had been seared in the flames of withering injustice. It came as a joyous daybreak to end the long native captivity. So it's over, and now we do our contradiction. But 100 years later, again, time is really specific. The contradiction begins with time, and what the problem is. The Negro is still not free. 100 years later, the Negro is still languished in the corners of American society and finds himself in exile in his own land. So therefore, we have come here today to dramatize the shameful condition. So you see how it set it up. It's just a normal ABT, which is it's really cool. Once you start seeing an ABT, you get examples of what the ABT is, you start recognizing it. The Cinderella has an ABT. The Gettysburg Address, I think, has an ABT. I Have a Dream is an ABT. The IMRAD is an ABT. Once you start practicing it, it's not something that you can learn all in one day, but once you start practicing it and recognizing it and using it on a regular basis, it becomes easier. So this is just an introduction. I know using the words and, but, therefore are a great setup to narrative and to build a story, but that can get a little repetitive. Therefore, there are other words to use. We have also, despite, so, equally, however, thus, identically, yet, consequently. So lots of different words that we can use that still follow the agreement, contradiction, consequence kind of following way. All right, so that is what I have on the ABT. Now I want to talk to you a little bit about my current research, and I will start with sharing you my personal ABT with you. So zooplankton make up the base of the marine food web, and they support the fish stock in Alaska's fisheries, which generates 12.8 billion in economic output. But zooplankton may be becoming smaller and less abundant as climate changes and oceans warm. Therefore, more resources need to be invested into using zooplankton as ecosystem indicators in order to predict the health of our fisheries. So this is why my research is important. And now I want to talk to you about what I'm specifically doing. Doctors David Kimmel and Jan Olberger recently received funding from NPRB to look at the effects of global warming on long-term changes in copepod size in the Gulf of Alaska around Line 8 and the Bering Sea around M2. And today I'm going to share with you how we're going about answering this question. So first, we started by identifying cold and warm years to begin our observations and obtain our preliminary results. We chose 2003 to 2005 to represent warm years and 2010 to 2012 to represent cold years. The next step was the collection of images. I searched the archive room that we have for samples of interest and then bring them into the lab for imaging. For each year, I collect images of 10 different species of copepod. Some are characteristically large and some are characteristically small. And for each species, I'll identify an image I'll identify and image 30 individuals at each of the 7 stages. So that's about 210 images each year for each species. And then I save each image with a specific naming convention that's later used when determining size. I also manually measure about 10% of all images collected at the microscope and save it in the file name. During an AI hackathon we participated in last year, we met senior solutions architect Ryan Simpson. And since then, we've worked together to develop a standalone MATLAB application. And this application automatically measures prosome length of copepods from the images collected under a microscope. The application starts by binarizing the image and turning it into black and white. And then it creates a mask, or it detects the object of interest, and in our case, the prosome of the copepod, which excludes the tail, the legs, and the antenna. It then filters the image, which is a method used to reduce the noise or enhance the edges of an image in machine learning. And then the application measures the identified prosome length in pixels. And then it converts the pixels to millimeters based off of a previously calibrated image of a micrometer at different magnifications. And the application itself determines the magnification based on the file name. So the two biggest obstacles, in my opinion, were getting the most precise calibration of image at each magnification and getting the algorithm to identify only copepods in the image. So this is a graph of an earlier iteration of the algorithm with outliers. The algorithm estimated copepod sizes incorrectly and it identified non-copepod targets. We observed an R-squared value of about 0.76, where each point represents a copepod and the dashed line is the 1-to-1 ratio line. And the blue line is the fitted linear regression line to the data. The human estimated length in millimeters was obtained from the 10% of the images that I manually measured at the microscope while collecting images. And the algorithm estimated length is just that. It's the length estimated by the algorithm. Now, this is the graph of a species Calanus marshalli with the outliers removed. It had an R-squared value of 0.92. So we can say that this algorithm is as good as a human and we're officially in business. We're now working with Brett Schulson, a principal application engineer with MathWorks, to refine and improve our image detection ability to reduce outliers and bump up that R-squared value. The plan is to ultimately create an algorithm that can detect and measure the length of copepod prosomes with the accuracy of a human and export the corresponding size data to an Excel sheet. But wait, there's more. In addition to length, we're also trying to develop an algorithm that measures the size of a lipid droplet. So this data can be compared to the work that Rika group is currently doing for us, which is exactly that, measuring lipid droplet size. The capabilities of our AI image detection algorithm is ever growing in theory for now. So goals for this project going forward are producing a MathWorks blog post about a private-public partnership in developing this application, to continue to expand the capabilities of our AI imaging algorithm so it can be used across groups here at NOAA. We're hoping that we're able to beef this up and pass it over to the ichthyoplankton team to do a little study on eggs. So yeah, we're really excited about that, and we plan on presenting our initial size results at the upcoming Alaska Marine Science Symposium. So I hope to see some of you there, and we hope to get some future publications out of this research. So this is all thanks to funding from NPRB. Thank you very much.
All right, we can clap. All right, thank you, Deanna. Our next speaker is Allison Jury. She's a fisheries biologist at NOAA's Alaska Fisheries Science Center. Her research focuses on early life history stages of fishes from the Northeast Pacific Ocean, Bering Sea, and the Arctic.
Okay, good morning, and thank you so much for joining me today. So I will be dovetailing off of what Deanna's already spoken about, and we're still going to focus on zooplankton, but narrow it down to a particular class of zooplankton known as ichthyoplankton. These are the planktonic stages of fishes, mainly their eggs, their larvae, but in some cases their juveniles as well. Similarly to what Deanna talked about in her ABT talk, we also contribute ichthyoplankton indicators to the fisheries management process here at the Alaska Fisheries Science Center. We do this through three main deliverables. The first being the ecosystem status report, which has a regional focus. The second being the ecosystem and socioeconomic profiles, which have a species and more specifically a stock focus. And then finally, we also are included as a set of considerations that all, in a risk table, that all stock assessment authors must fill out that details if and when changes are made to the acceptable biological catch that they recommend. However, for today's story, I want to focus less on how we apply ichthyoplankton data to studies of the ecosystem and talk about how we, how we actually identify them in our samples. So for those of you that aren't familiar, when we collect plankton, we bring it back to the lab and it comes into a plankton jar similar to what's pictured here. And unfortunately for us, they don't come pre-sorted. That would be really lucky. So what we then have to do is process the plankton sample and remove out the early stages of fishes. And just to remind you, that includes their eggs, larvae, and juveniles that are associated with plankton. So what we get from this sorting process are individuals that we've removed from the sample in these rough groupings. And we now need to classify them into taxonomic groupings, preferably into species, but that's not always the case. But the question is, how do we actually do this? How do we assign these individuals into species, considering that they look very different from the adults due to their small size and adaptations to surviving at small sizes in their environment? Well, we use a combination of tools. Firstly, we start with microscopes that allow us to magnify the individuals and highlight their features that we can use to identify them, that distinguishes them from other individuals. We also use a combination of laboratory guides and illustrations to assist us when we're looking at the microscope. What's really neat about being in the ichthyoplankton team of Ecofoci is that back in 1989, we literally wrote the book that sets the baseline information of all of the taxa that we currently are able to identify in our large marine ecosystems. Within this guide are illustrations organized into what we call developmental series, which this example picture here is for walleye pollock. And a developmental series are sets of illustrations that highlight progressively later stages, and they highlight the key characteristics that help us distinguish one individual from another individual in a different species. Now, for walleye pollock, this is a pretty complete developmental series. We have an illustration of a late stage egg, a newly hatched larvae, through progressively larger larval stages and into the juveniles. This is not often the case for many species. We have what we call gaps in the developmental series, where certain stages we don't have yet descriptions for or illustrations. Therefore, the mission that we conduct as part of the ichthyoplankton team is to continue advancing our taxonomic resolution, our ability to make reliable species level identifications. And we do this through continued plankton monitoring of our large marine ecosystems, which include the Gulf of Alaska, the Bering Sea, and the Arctic. What gets really exciting for us is when we get to sample either a new ecosystem or an ecosystem that we monitor but at a different time of year, because that provides fresh material of potentially species we don't often encounter, as well as stages we don't see during our normal monitoring efforts. And what I have here are a collection of illustrations that are part of a recently submitted manuscript that put forth some advances that we've made into our developmental series of several species. And we've been able to fill gaps in the developmental series of these species, either by replacing poor quality illustrations with newer, better quality illustrations, as well as in the case of Arctic cod, adding a stage that was previously undescribed and not illustrated. Now, once we have these gaps filled in the developmental series, we then contribute them to our laboratory guide. But unlike back in 1989 with a static guide, we have migrated this to an online format known as our ichthyoplankton information system. And what that does is it allows us to disseminate widely these taxonomic advances, as well as in a timely manner. And that contributes to our ability to present reliable species level data in our area in a timely manner that produces robust data sets that then can be contributed to support sustainable fisheries in our area and examine questions of community resilience under climate change scenarios. And with that, I'd just like to thank the co-authors, the Plankton Sorting and Identification Center in Poland for all of their hard work, and you for your attention.
And next we have Ned Copeland, also known as the Saildrone Data Guru. He is a physical oceanographer at NOAA's Pacific Marine Environmental Laboratory. Ned advises on saildrone instrumentation and the variables they measure, among other things.
Thank you. So this is not much of an eco-foci topic. It's about wave measurements from saildrones, but I volunteered to be one of the guinea pigs for the ABT and the first seminar this year. So we have these saildrones right here. They've been developed in 2015. They used to look like this, taller than the present one, but in 2021, we, working with Saildrone Incorporated, made smaller Saildrones with lower wings and more rugged. And we put them out in hurricanes in the Atlantic. So this is the camera on a Saildrone during Hurricane Sam in Atlantic in September 2021, September 30th. And you can see it's a place where you really wouldn't want to be yourself if you're out there because it's pretty rough. Not a great place. You know, I always thought maybe we should just send NOAA officers out in Zodiacs, but that's probably not a good idea. So we put them out. We measured the. This is a map here. This is the hurricane track right here, and this is a circle here of the closed pressure contours. And various contours of velocity as you finally get to the center of the hurricane. And this is at the time when the hurricane was closest to the Saildrone. The Saildrone is that little symbol right here near the center of the hurricane. At the same time, well, before that time, there was a NOAA data buoy over here. The hurricane passed by the NOAA data buoy and arrived at the Saildrone. What I'm showing here is the wave spectrum first measured at the NOAA data buoy in green and the Saildrone in red. What this really means is this is time here in September, October 2021. This is the height of these are the wave heights of the significant wave height of the waves. And then try here. So this is the significant wave height is the height of the highest, the average height of the highest 1/3 waves. But the Saildrone itself measures individual waves, and these are wave profiles here of 15 minutes. I don't know if it's over here with the mouse. There we go. 15 minutes of record here of waves, and you can see that this wave right here is 27 meters high. That's a pretty big wave. And if you looked last summer at the literature and the newspaper, there were some people in British Columbia who put out measurement buoys to measure waves, rogue waves in the Pacific, and they measured what they thought was the highest wave ever measured. Well, we also measured, that was 27 meters. We also measured a 27-meter high wave here in the hurricane. There's a difference though. They were looking at rogue waves, which just appear out of nowhere, not in a storm. Well, these are measured in a storm, so there's a difference in what's going on. Okay, great. So we measure these waves, but how good are the measurements? So a normal wave measuring device like this here is a wave buoy. This is the same wave buoy that was near the Saildrone. And you notice that they're circular, they're discus shaped. But some wave buoys are actually pointy. They look like they're ship hulls. And our Saildrones are also pointy. They look like ship hulls. So this is the standard way you measure waves, but this is the way we're measuring waves with our hurricane. So how good is that? Well, one way to determine that is to look at what's called the wave spectrum here. And I'm going to show you a series of plots. This is the spectrum of the waves measured at the Saildrone, and this is the spectrum of the waves measured at nearby NDBC buoy. And basically what this says is we get time down here on the x-axis and energy of the waves here. So the more energy, sorry, frequency of the waves here, and then the energy is in colored bars here. And the more energy there is, the bigger the waves are. And you can see here that the spectral peak of the Saildrone found at Saildrone and the spectral peak found at the NDBC buoy. And this is the difference between the two. So first off, I just want you to notice that they look pretty similar to each other. So the Saildrone and the buoy, basically seeing the same kind of wave field here in a statistical sense. So how good is that in general? Well, we had times where other Saildrones sailed near NDBC buoys kind of fortuitously, but sometimes we actually did an experiment. So I'm just going to show you some of those results here. So always there's going to be the sail-drone spectrum on the left and the buoy spectrum on the right. And it's just a visual comparison. That's really much to see the differences down here, but that's not so important. So you can see that in this instance here, the sail-drone was seeing much the same thing as NDVC buoys there. It's giving us good confidence. And the difference here is I can't quite see this over here. The water depth is only 30 meters here, and there we go. Water depth is 30 meters, and the sail-drone and the NDVC buoy were a kilometer apart. But the catch is the sail-drone was out in deep water because you couldn't bring the sail-drone in at 30 meters of water because it could run aground. So it's a comparison, but it's not quite the right comparison. And I should have said in the earlier one, where the sail-drone was measured with the NDVC buoy, they were about 200 kilometers apart. So it's not a good check on the ability to measure these two things. So if we go down the list here, here's a case where the sail-drone now is only a couple kilometers apart, but still in shallow water, this buoy. And you can see though, visually, they're much the same thing. The two spectral peaks are about the same. The buoy is seeing a little bit higher waves than the sail-drone. And again, another case now, they're still close to each other at 4 kilometers, but they're still in fairly shallow water. So the buoy's in shallow water, but the sail-drone's in deep water. The waves change as they come into shallower water. So it's really not a fair apples-to-apples comparison. But you can still see the spectral peaks are about the same, and the look of the spectrum is about the same. Now, also within 3 to 10 kilometers and still shallow water, but again, we're seeing the same phenomenon both places. Here, also a few kilometers away, but now we're in deep water. So now the sail-drone is seeing this, and the wave buoy is seeing that a little bit different, but still visually much the same impression of the wave spectrum. And now here's a case where they look basically the same, but these waves are really tiny here in August of 2021, and it was out in deep water, but there was no real storm going on during this time. But the sail-drone happened to be by the buoy, so we use these measurements. And last case here, again, visually very much the same. The waves, the buoy saw a little bit higher waves than the sail-drone did. One more case, again, visually the same. So the conclusion is, how well can sail-drones measure waves? Well, reasonably well, but precise validation requires longer observations of different wavelengths and directions. So if you're really going to use a sail-drone as a substitute for a wave measuring device in NOAA, you'd really want to do some tests where you put the sail down for several months at a time, sail around a buoy with all kinds of different wave conditions, storms, no storms, the sail-drone going in different directions because it is pointy, it doesn't respond the same way, but that's something yet to be worked out in the future. And let me just go back to, this is the slide that spurred this whole thing going. And you can see, by the way, if you subtract these two spectra, you'll see that the waves of the sail-drone is higher at this frequency and lower at that frequency. And that's because you can see the spectrum has moved down. The waves have actually changed in this 200-kilometer shift from one place to another. The waves went from the buoy here to the sail-drone. They got lower frequency, and that's because the wave shape bill is developing. Anyway, that's the end of my talk.
Yeah, that is all for our speakers, and now we have some time for questions and discussion. If the speakers in the room want to come to the front, sorry, Ned.
I'm back.
And then Deanna's going to join us online.
Hello.
And we have one question in the chat for Ned. What is the benefit of a sail-drone versus a buoy?
The benefit of the sail-drone versus a buoy, first off, the buoys are maintained by the NASA Data Buoy Center, and they are rather expensive to keep out there and to maintain, and sometimes they fail. And this year, 2 of them failed in the Gulf of Mexico, and they used our sail-drones as substitutes for their buoys to see if they could use a sail-drone to make the measurements. The sail-drone does other things besides that the buoy doesn't do. It makes more measurements in the water and in the atmosphere than the buoy does. and it has the ability to move around, which we were using the hurricane to move out just to intercept the hurricane, which that buoy doesn't do. But that's kind of the difference between the two. Thinking about perhaps using sail drones in some places where they have buoys now.
Do we have any other questions in the room?
I have a question for Ned. So is the,
Can you come up to the front? Just, we're doing the hybrid thing and we want people at home to be able to hear. Thank you.
is that close enough?
Yeah.
So my question was about, is the difference between the buoy's ability and the sail drone's ability to record this data, the motion that the sail drone is under?
Probably it's the fact that it's got it's pointy, it's got a longer, the hull is longer than it is wide. I don't exactly know the reason behind it. And, you know, we don't really know which is the better measurement, although the NDVC buoys with their viscous shape have been around for a long time, so that's kind of the standard. So it's probably the fact that the elongated hull is responding differently to the waves than the viscous is. And again, that you really require a months-long measurement with the two interacting together the whole time. That's something to happen down the road. But here we're just trying to, the very first time, trying to get some idea, do they measure waves at all? And the answer is yes, and pretty well.
Great.
We have a question from the chat from Frank Hernandez regarding the MATLAB app. Are there preservation issues that affect the FID drop?
That's a great question. We haven't gotten to doing the lipids yet, but the answer is yes. Thank you, Dave.
Yeah, Sean?
Yeah, I'll come home.
Thank you.
The camera's also that way.
Wants to see me. My question's for Ali. So I hear all about the magical facility that is Poland, but I have no idea what they're actually doing there in order to take that lovely jar of slurry and to get some distribution of fishes, I'm assuming. So it's kind of two-point question. One is, can you elaborate just really quickly on what Poland's doing? And two, is there an instance or an application for Deanna's work with imagery to help improve the data that's coming out of Poland's laboratory?
Yeah, thank you. Excellent question. So yeah, as Sean alluded to, so those of you who are not familiar, we are really lucky at the Alaska Fisheries Science Center, along with the Northeast Fisheries Science Center and the Southeast Fisheries Science Center, in that plankton jar that I pictured, we don't actually process that in-house, which saves a lot of our manpower because that means we can focus on quality assurance and research when we get the data back. So what happens in Poland is once we come back from the boat, we have our boxes of jars. We send all of that over to Poland. And there is an enormous team of both zooplankton and ichthyoplankton ecologists. And they go through and they sort through the plankton samples. And so from our side, they first do the processing for ichthyoplankton, and they'll remove all of the ichthyoplankton that they find. They will also do some resorts on a subset, and if they find more than 10% of the ichthyoplankton was missed, they resort it again so that we have as good as possible quantitative data from that net. And we've also collected measurements of how long the net was in the water, as well as how much water was filtered, so we can get a quantitative assessment for that chunk of water we sampled, how many fish eggs, larvae, and juveniles were collected. That same process gets repeated for zooplankton, except they don't do the whole sample, they do an aliquot because there's a lot of zooplankton and nobody wants to count 10 million copepods. We can extrapolate that. So once they have the ichthyoplankton separated from the sample, they then run through and they do those steps that I detailed. They use laboratory guides that we generated, as well as auxiliary help with developmental series and illustrations we send to them. They do all of the identifications, they enter it into our database, and it's this amazing contraption that comes back that Kimberly, our database manager, ensures everything looks good. And then, like I said, we follow up with quality assurance, and There's a lot of taxa that they do really, really great. There's some taxa that they don't do really, really great, and that's partly because we don't yet have great diagnostic tools to help with that. There's still questions on how species are related to each other, what they actually are to that level. Going back to the second part of your question, do we see imaging as contributing to that? Yes. The one issue that I see with that with ichthyoplankton is we rely on pigment characters. And so that gets a little bit difficult to look at the resolution we want for a management perspective. Like we can throw out an imaging system, and I've worked, I see Frank's on the call, with Frank Hernandez back in the Gulf. We had what we call ISIS, the in situ ichthyoplankton imaging system. It's towed behind the boat. It gets wonderful pictures. They also fish at Oregon State University and several other places. But the issue you have is you don't get the same taxonomic resolution. Some images you can classify as species, but what I fear for our ecosystem is the species we want to know a lot about are the gadids. And we need to be able to identify Pacific cod from walleye pollock. An image won't necessarily get you that level of detail because you have to have the right orientation to know if the pigment patterns are that Pacific cod or a walleye pollock. So that's one of the limitations on the larval side. But where I see the imaging really being advantageous for us is co-opting our egg database. We are the only center that identifies fish eggs, or asks Poland to identify fish eggs, let me be honest. And it's really, really cool. But we don't really do that much with the egg data. And part of that's because how we identify the eggs for a lot of the times is we have a diameter range. If an egg falls into this diameter range, it's at a particular season, it's a walleye pollock. If it doesn't fall in that diameter range, it's a different species. What we're not tracking are individual lengths of the eggs, and that's where I see the imaging coming in, is that we can use that algorithm to assist in the sorting of the eggs. We can take a picture of eggs and bloop, bloop, here's the quantitative data. These ones are in this diameter range, therefore they're pollen. But we can also then get lengths or diameters of those eggs, and then we can start to ask ecological questions. Is egg size changing over time as climate warms? There's been work on gadids in the Atlantic that shows relative size of the egg relates back to the health of the spawning stock. We don't have the ability to ask those questions right now, but with advances in the imaging and having an algorithm generate those diameters, I think we can move that way. And that's really where I see the advantages for the imaging. So thank you.
Couldn't have said that any better. Thank you, Allie.
So I have a question for the imaging. Is that shipboard, or are there any possibilities or technologies for doing that on mobile platforms?
Yeah, the goal is that it will be used on live animals on board a ship. But that is, we're starting with in situ or ex situ under the microscope images to train the algorithm so it can identify it, and then hopefully we'll be able to we have an RZA that we take rapid zooplankton analysis on board, and hopefully we'll be able to just take a picture of an aliquot and use the algorithm to automatically identify which species are being present based off of the images we've trained it on from Poland, who is collecting images for us, as well as the images that I collect from our archive room. And then it should be able to tell us what's there and how big it is and everything like that. So that way we don't need to send a zooplankton ecologist on every cruise. Anyone can do it, just snap a picture, and this algorithm should be able to do it all for you.
Any other questions from online? For any of our speakers and in the room? What's the time outlook for these technologies? I guess for either of you, for being able to build up that egg database, and is the technology development coming out of Deanna's research that you would use for that? And do you have historical archives of eggs that you'd be able to apply that to, or is that a going forward?
Yeah, so great question. So I'll jump in first, Deanna, and please correct me if I'm missing something. So short answer is yes. We are going to co-opt all of the amazing work that Deanna and Dave have been doing because they have already laid the foundation. And the nice thing is eggs are spherical, so they're pretty easy for a computer to be like, oh, I found an egg. So that part we're really excited about. How I envision this moving forward is we're in a marathon. It's not like a short race. We're not a 100-meter dash, but it's a marathon. So the first step is that Poland typically puts all of the eggs at a station back together. The first step was requesting them to not do that and keep them separated by taxa, so by species. And so that gives us the ability that they have the eggs pre-sorted and they stay sorted. And then we can start next year, we're going to add the imaging for the eggs, is my goal. So this year they're starting to take some pictures of ichthyoplankton. I want that so we can throw that onto our online ichthyoplankton information system and additional line drawings, have beautiful pictures. Going back to your question though about like what material we have available, those of us that do collections type work tend to be hoarders. So we actually have all of the ichthyoplankton that we've ever collected. And we have them maintained both in-house in our ichthyoplankton lab. We're always happy to show people what we have. And then we've also had a long-standing collaboration with the University of Washington, and they do the curation of all of our historical samples, so 7 years and beyond. So what I'd like to see is that become a thing that we start and then move forward and becomes a standard operating procedure is part of the vision, but also we have the ability to go back and look at historical samples. I'm not necessarily thinking we're going to image every sample of egg, but do approach similarly to what Deanna and Dave did and highlight certain years or certain surveys and get some of that coverage so we can look in the past, but then make that this process moving forward.
Yes, absolutely. And the application it's working, it's generating data. So really all it is imaging the eggs or plankton or copepods or whatever it is you're looking at specifically. So yeah, it's working. And we have 2 developers that are working on it. So we're still continuously refining it and doing tweaks and bugs and stuff like that. So it's something that's ever moving.
Last chance for one more question. All right, thank you, and thank you all the speakers. We look forward to seeing everyone next week at the same time. We'll have 2 speakers, Emily Hayden and Jens Nielsen, and these 2 presentations in combination will provide an overview of ocean temperature and sea ice variability, paired with the discussion of its potential impact on phytoplankton bloom timing and zooplankton coming out of diapods in the Bering Sea. So we'll see you next week.
Thank you.